Hello, my name is Fadi Frischknecht, and I will present you a paper from my lab written by Catherine Moreau, a former PhD student who also did most of the work together with several colleagues of hers in the lab in Heidelberg, but also in collaborating labs in Heidelberg and elsewhere. This paper talks about a very unique interface between the actin-binding protein Profilin and actin itself that we found to be important for the motility of the malaria parasite. You can read more about my work on our website, sporozoid.org. You can also follow me on Twitter. Now, motility is important at three different stages of the malaria parasite life cycle. The one being the merozoite getting into the red blood cell, a very quick encounter. One being the motile egg, the ookinete, traveling across the midgut epithelium inside the mosquito gut to establish an oocyst. Within this oocyst, then sporozoites are forming, and the sporozoid is the most versatile motile cell of the plasmodium because it needs motility to get out of the cyst, it needs motility to get into the salivary gland, to disperse within the salivary gland. When it's spat into the skin of the vertebrate, then it needs to migrate inside the skin to find a blood vessel, and it needs to migrate again to come out of the blood vessel and enter into liver cell. Plasmodium sporozoites are slightly curved cells that move in a circular manner on a flat substrate, so you can very easily make very long movies and quantitate very many sporozoites at the same time. They move at 1 to 2 to 3 micrometers per second. So they're very, very fast, about 10 times faster than neutrophils, which are the fastest cells that migrate in our own body. Now, this motility is based on actin filaments, the turnover of these actin filaments, and the actin filament binding motor protein myosin, which moves actin filaments backwards, or so it is thought. The actin filaments, we assume, are formed at the front end of the sporozoid, and the myosin is then moving them backwards, and the actin filaments are linked via a number of transmembrane proteins such as TRAP to the substrate, and that ultimately gives the parasite the push forward. Now, the actin filament is highly dynamic, and that means that dynamic is regulated. It's partly regulated by the structure of the actin molecule itself, but it's also likely regulated by the binding of actin-binding proteins. One of the most famous and one of the most conserved ones across eukaryotes is Profilin. And Profilin is thereby nearly ubiquitous actin-binding protein that binds actin monomers, and it can help actin monomers to exchange an ADP for an ATP and hence make them available for polymerization. The curious thing in Plasmodium profilin is that it has an additional domain that most other profilins do not have. And this domain contains 3 acidic residues, E, D, and E, which are binding hydrophobic stretcher. They seem to bind a hydrophobic stretch on the actin monomer, as you can see in the 2 types of inverted structural models of actin on top and profilin on the bottom. So when this structure was resolved and reported in a paper by our collaborator Ina Rekursala in 2008, she speculated that this binding must somehow contribute to the stability of the protein complex. And in our paper, we now want to investigate whether this is the case or not. Our first set of experiments was to model this dynamic interactions in silico in a computer using a process called molecular dynamic simulations. So essentially, you put the crystal structure data into your computer, you put the proteins close together in the conformation you know from the crystal structure would roughly fit to their natural connections, and you let this simulation run for about 150 nanoseconds. It's very short interactions. And what you can see on the left-hand side, again, the position of the EDE acidic amino acids at the top of the sort of arm extension of the Profilin. And when you let the simulation run, you can see that in red and blue where the proteins are changing a little bit, and there is not much change in the positioning of this arm-like extension from the profilin towards the plasmodium actin on top. If you look then at a mutant profilin where the EDE was exchanged for a Q and Q, this is the middle complex, we actually don't see much of a difference just looking at the simulations. However, if we change them to apolar AAA amino acids, so 3 alanines instead of the glutamic and aspartic acids, then suddenly we observe that the arm cannot be kept in space. There is a critical hydrogen bond between the profilin arm and the actin monomer that is lost, and hence the arm just wobbles around, suggesting that there is a weaker interactions between the profilin arm and actin in this AAA mutant. Now, to see whether this is really true also in biochemical experiments, we purified these 2 proteins, profilin from the parasite, and we used in a first assay actin from the muscle of a pig. Actin are highly conserved, so we thought this was good enough. And the experiment we performed is that we let actin polymerize in a test tube just by itself, as you can see here on the very left, in the very leftmost graph and plot. So the top line, the light blue line, corresponds to actin polymerizing over time in the absence of profilin. You can see here there is 0 micromole profilin present. Now, when we add increasing concentrations of profilin, here exemplified at the 10 micromolar level, you can see that actin is not capable to polymerize as efficiently anymore as before in the absence of profilin. And this can be seen either by the lower relative initial rate, as you can see in the below graphs, or in the flattened curve on the top graph, where you measure the fluorescent intensity of the formed actin filaments. When we look now at the mutant, the Q and Q mutant, if you compare the curves and also the graphs towards the natural profilin, you see that it's already got a weaker capacity to interfere with the inhibition of actin polymerization. And in C, with the AAA mutant, it seems to be even stronger. So if you add profilin that has these triple A mutations, it barely interferes with the capacity of actin to polymerize. So it seems that both mutants show weaker actin binding in vitro, with the AAA mutant being even weaker than the Q and Q mutant. You might wonder, why did we use pig actin and not plasmodium actin? And the reason is that it's much easier to purify pig actin than plasmodium actin. But our collaborators in Norway and in Finland at the time, they were very persistent, and they finally managed a method to purify plasmodium actin as well, and could do the same experiments again. And essentially, you now see the same thing. In A, you see that increasing concentrations of plasmodium profilin also inhibit the polymerization capacity of actin. In B, you see that the mutants interfere with the capacity of the diminished actin binding of profilin. Here, however, you do not notice any difference between the Q and Q and the AAA mutant. On panel C, you see a different type of experiment where you let actin polymerize, and then you spin down the actin filaments, and you look how much of the actin you can retrieve from the pellet, which is the filamentous actin, in dark, or from the supernatant, which you can see in white. And again, here you can see with increased concentrations of profilin, you get less of a pellet, you get less actin filaments formed with time. And if you use the mutants seem to be again in between the situation with plasmodium profilin and just the control where actin polymerizes by itself. So again, showing that both mutants show weaker actin binding in vitro. In this case, about the same level. So with these results, we felt motivated to now go into the parasite and introduce these mutations in the genome of the parasite. And we used the Plasmodium bergii rodent model of the malaria parasite, and we introduced, or actually replaced, the Plasmodium bergii profilin with the Plasmodium falciparum profilin. This had absolutely no consequence of the life cycle of the malaria parasite. The merozoites were invading red blood cells just fine. The ookinates invaded mosquitoes just fine. The sporozoites, as you can see on the left, they were gliding just fine on the flat surface. On the bottom, you can see a time lapse of one parasite moving in circles. And on top, you see selected tracks of parasites that have moved for a certain time, and you see most of them move in nearly perfect circles for many, many minutes. Curiously, when we now looked at the mutant that showed the Q and Q mutations, we couldn't see any difference. While when we looked at the AAA mutant, there was almost no capacity to move consistently on a circular trajectory. So we found an aberrant motility of the AAA mutant, but not at the Q and Q mutant. We also quantified this motility in a different way. Here we looked how the parasites were capable of moving at all. So we looked at all the population of parasites that were apparent in a number of experiments, and you see the numbers here is in the hundreds of parasites that we've looked at. And on a normal Plasmodium bergii rodent model sporozoid assay, we can see about 50% of the parasites moving in a persistently forward-moving manner. This is the dark part of the bar. While when we actually exchange the endogenous Plasmodium bergii profilin for the Plasmodium falciparum profilin, it's the second bar, you see that already less parasites are moving consistently. Yet there was no difference between the numbers moving in a consistent, persistent manner in the Q and Q mutant, while we saw very few AAA mutants moving in a persistent way. Persistent means for a long time. So when we make a movie for about 5 minutes, how many of the parasites are moving for the entire 5 minutes? Very, very few of the AAA mutants were capable of doing this. Now, curiously, if you look to the right, you can see that the wild-type parasites of the rodent malaria parasites, they move at just under 2 micrometers per second on average. While if we put in the Plasmodium falciparum profilin, these mutants suddenly moved faster. But remember, from the left graph, they actually seem to move less persistent. So they're not very good in moving for very, very long. So they're not very great marathon runners, yet they're very fast. So they're probably very good at the 400-meter track, but not at a 24-kilometer track. This corresponds a little bit to the observation that we see at higher temperature. At higher temperature, fewer parasites are moving persistently, yet they move at a faster speed. Now, important is that we compare the Q and Q mutant to the Plasmodium falciparum wild type, and we see no significant difference in their capacity to generate high velocities. While the ANA Plasmodium sporozoites, they really were much slower in comparison. So not only were they not very good in running a marathon, but they were also not very good in doing a sprint. Having said that, not very good in doing a sprint, they're still moving at 1 micrometer per second, which is, again, about 10 times faster than the neutrophil moves in our body, and the neutrophil is the fastest cell of our own bodies. So having said that they move slower, they still move at a remarkably high speed. Here is just a highlight again to show you that how few of those are actually moving, right? And here, that few of them, those few that are moving, are actually moving at a slower speed. Yeah? So lastly, we wanted to understand whether there is a difference in the capacity of the parasite to generate a force, which is of course important for generating motility. And we previously established a very sensitive assay which relies on the use of optical or laser traps. So in this assay, we take a microsphere, a bead, and place it on the surface of the parasite. And we then measure two things. First, the capacity of the parasite to drag that bead out of the laser trap. This gives us an indication about the force the parasite can apply. Secondly, we measure how quickly the parasite is able to translocate this bead to the back. This gives us an indication of how fast the so-called retrograde flow is that is very likely mediated by myosins moving the actin filaments backwards. And this gave us a very surprising result. So here we found in panel B, when we looked at the overall capacity of the parasite to generate force, that both the wild-type Plasmodium berghei parasite and the Plasmodium berghei parasite that expresses the Plasmodium falciparum profilin can exert about the same level of force. Yet both mutant parasites, those expressing either the QNQ profilin or the AAA profilin, they have much reduced capacities to generate a force. And what here is indicated is the percentage of parasites that manage at a certain laser trap strength, which is 70 piconewtons or 130 piconewtons, to rip a bead out of the laser trap. In a second experiment looking at the speed of the retrograde transport, we found exactly the opposite. So here, the wild type and the Plasmodium falciparum profilin expressing parasites show a very rapid but rather lower speed of around 5 microns per second as a maximum, while the two mutants could show speeds between 10 and 15 microns per second. So in just less than half a second, a bead would travel from the front to the parasite to the back. It's an incredible flow on the surface of a cell. It's actually faster than anything that has been measured on any cell before. And this is just quantitated on the very right graph over many different observations. Now, that suggests that the parasites that express these mutants profilins cannot generate a lot of force, yet somehow they can produce an incredible speed with which they can pull the beads backwards. How that goes together now is kind of what we try to illustrate in this rather complex cartoon at the end of our paper, which honestly is just an admission that we're confused at a higher level. We've done a lot of experiments. We found out how Plasmodium berghei can be modified in its way of generating force by expressing these mutant profilins, how the mutant profilins interact with actin. That's all very good, very solid, very quantitative data, but to understand how the parasite moves forward, it really leaves us with a still very, confused model system, confused model. So we think that at one level, there is actin modulators. There is proteins that polymerize actin filaments. There is proteins that make these actin filaments bundle into larger, maybe, structures, cross-linkers. There is molecules, proteins that disassemble actin filaments. So we could call this the actin state. The actin state describing how many actin filaments are there, at what spatial side of the parasite, how they're oriented, and how dynamic they are. And this influences the retrograde flow on the surface and the force with which the parasite can pull on a substrate in opposite directions. And this must somewhat be linked by a factor we don't really know. This could be a protein, this could be a set of proteins, this could be a type of membrane flow that we don't know, maybe coming from exocytosis at the front end as the parasite moves. But all this conspires together to produce motility. And we think it's so complicated because this complexity gives the system a level of robustness that even if we disturb these actin-profilin interactions, and we can measure this very precisely in biochemistry experiments and in laser tweezer force experiments, we don't see a difference as the parasite progresses through its complicated life cycle. Motility is likely so important that you can actually introduce quite a few mutations that have no effect on the overall outcome. Now, I hope that you enjoyed that talk, and if you're confused at a higher level, maybe that's not a bad thing after all. Thanks for listening.